Yeah, so I will share this share the screen. Is the screen visible to everyone? Can anyone reply? Okay, thanks. So in this class, in the fourth, this is session 5, okay, regarding the course Cell Biology, Cellular Organization, Division and Process. And my name is Kundan Solanki. I am a Prime Minister Research Fellow in Indian Institute of Technology, Indore. Okay, and so today in this class, we'll be discussing about processes of meiosis. Okay, so in previous class, we discussed about mitosis. And in this class, we'll be discussing about the meiosis uh, process. Okay, we'll be starting with the basics of meiosis and how it occurs, what are the different stages in the meiosis, and the regulation part of the meiosis. Okay, the different at the different stages of the meiosis, what are the regulations which are happening, and what are the key points to be noticed. Okay, so we'll be seeing that thing, and if there is a stoppage in the lecture, so then I will try to again rejoin. But if it is persistent internet connection issue, then we can reschedule. Otherwise, let's see. Okay, currently how much we can cover. So these are the two process, mitosis and meiosis. Mitosis, we already discussed the various stages of the mitosis and the regulation at each and every stages. Okay, there is G0 phase, then G1S, G2 and M phase. These are the four different stages, and each and every stage is a specific regulation occurring by the protein and protein kinases and cyclins. Okay, and various other post-translational modification occurring on the protein which help mediates its activation. So, so at this that we looked at this thing, okay, in mitosis. And in meiosis, we'll be talking about so this is generally the diploid cells that is 2n in number, which is in G1 phase. Okay, it start accumulating certain proteins and all the other machineries, replication machineries, which gets activated in the S phase, which is a DNA replication stage, and the chromosome number gets doubled in this case, and then the segregation of sister chromatids occur in the anaphase stage. Okay, and then the diploid cells are formed. So after the segregation, so this is the process of mitosis. In meiosis, the diploid cell is actually formed, and cells is first diploid, and after the DNA replication, the chromatids are recombined. Okay, just a second. Hello. Is the voice proper now? Actually, there is little internet issue. So, is it audible currently? Okay, so we will see if the problem persists. We will try. Okay, 5 minutes. If the problem remain persistent, then we can reschedule this lecture to other day. So please let me know uh, if there is any issue in the audio or video. Yes, so I was talking about process of meiosis, and in meiosis, generally, the diploid cells are replicated in the S phase, and there are two stages in the meiosis compared to mitosis, in which the DNA replication occurs and then the sister chromatids get separated. So there is only one stage involved in the mitosis, but in meiosis, there are two different stages, that is meiosis 1 and meiosis 2. So in meiosis 1, the sister chromatids are replicated in the DNA replication stage, and then in the prophase stage of the meiosis 1, the sister chromatids, the chromosomes, homologous chromosomes, are paired with each other to form a homologous pairing, okay, by the synoptimal complex, which we'll be discussing further. So homologous pairing occurs in the prophase stage of meiosis 1, and the process, then the exchange of the genetic information occurs between the homologous chromosome during the prophase stage, specifically the diplotin stage, the chitin stage, sorry, that we'll be seeing further. Okay. So the main purpose of meiosis is to form the homologous pairing and then to share the genetic information between the two homologous chromosomes. So, and that is the reason that in the meiosis, generally we get the genetic variations in the offsprings. Okay, so meiosis specifically occurs um, in the reproductive cells in the body. Okay, in the ovum as well as in the spermatocyte during the spermatogenesis. Okay, and uh, due to involvement in the reproductive the fertilization process. So because of the involvement in the fertilization process, that uh, the chromosomes are generally in the end phase. Okay, so there is a monomer of the chromosome side. So in the meiosis one, the pairing occur, and then the genetic informations are shared in the prophase stage, and then again in the meiosis two, the sister chromatids are separated from the homologous chromosome, every individual chromosome. So from the one diploid cells, you can get four haploid cells. Okay, so this is the process of meiosis, and there are two stages involved in the meiosis process. So let's discuss a little bit in detail regarding how the meiosis occurs. Okay, the different stages in the meiosis. So there are as was discussed in the mitosis. The same process occurs in the meiosis as well, but the stages, that's there, involvement, there are two steps. Okay. So in the G1 phase, the cells are diploid, and the cell generally accumulates certain protein machineries and replication systems to undergo the replication process in the S phase. And in the S phase, here you can see the centriole divisions occur, as we discussed in the in the mitosis as well. The division of centrioles occurs, as well as the chromatids are also replicated by converted into a chromosome, okay, pair of chromosome. And in the G2 stage and G2 and the prophase stage of meiosis 1, the sister chromatids are the chromosomes are homologous chromosomes are paired with each other, as you can see over here, and the centrioles also starts assembling the microtubules. Okay, and it starts organizing the microtubules to align the homologous chromosomes at the equatorial plate in the metaphase 1 stage. So in the prophase, generally the pairing of the homologous chromosomes occurs, and the important stage is in the meiosis, that is crossing over of the genetic information between the homologous chromosome occurs in the prophase stage. And in the metaphase stage, after the crossing over has occurred, the spindle fibers aligns the chromosome in the equatorial plate by the help of the centrosomes at the opposite position, by placing them at the opposite position. And in the anaphase 1, as you can see, the homologous chromosomes are separated through each other, okay, by the spindle poles. And in the telophase and cytokinesis 1, you can see the two homologous chromosomes are separated, and the nuclear membrane, nuclear assembly has occurred. Okay, and the two sister chromosomes are separated by each other. So over here you can see the growth meiosis one is specifically known for the variation in the genetic information which it provides. Okay, in the mitosis there is no exchange of the genetic information between the two sister chromosomes, two homologous chromosomes, but in meiosis one there is a exchange in the genetic information at the point known as chiasma. Okay, so the point at which the genetic exchange occur between the chromosome is known as chiasma, and due to this reason there is a variation in the genetic variation between each offsprings of the individual parents. Okay, and there is a genetic variation in each and every individual. So uh, it is the reason genetic uh, crossing over and genetic crossing over is the reason behind that. So after that, in the at telophase and cytokinesis, further the processes occurs now of the meiosis 2. Okay, and here you can in the crossing over stage, actually the point we need to discuss is that the genetic variation, as I told. So the homologous pairing and the formation of chiasma and genetic information exchange occurs. So this is one possibility. There can be many other possibilities as well. So here you can see that this chromosome and the black chromosomes are in the one zygote, one cells. Okay, but there can also be a possibility of this green chromosome with a pink exchange genetic information with this chromosome, as you can if you can see my pointer. Okay, so these two combinations can also be there in a single cell, and these two combinations can also be there. Also, the genetic information which was exchanged can also be at a different point. Okay, so these multiple combinations occur, and that is why the genetic variations are generally found in the meiosis 1. In the meiosis 2, after the crossing over has occurred, the centrioles, again here you can see are disintegrated again. Okay, and in the meiosis 2, in the prophase 2 stage, again the centrioles are aligned, microtubules are formed. Okay, around the centriole, and here you can see again in the metaphase 2 the chromosomes are aligned with each other, and in the anaphase 2 the sister chromatids are separated, as was seen in the mitosis stage of the cell cycle. Okay, so meiosis 2 almost replicates the mitosis of the mitosis stage of the cell cycle, except the point that in after the mitosis process we get a diploid cell, but in the meiosis, after the stage of the meiosis 2, we get a individual haploid cells. Okay, so this is the difference between the mitosis and the meiosis 2. Otherwise, the process remains the same. Moreover, in the meiosis 2, The genetic information which was being exchanged in the prophase 2 stage, prophase 1 stage of the meiosis 1 does not occur in the prophase 2. So in the prophase 2 stage, the spindle assembly and the attachment of the spindle poles to the centromere occurs, but the genetic information in the crossing over only occurs in the prophase 1 and not in the prophase 2 of the cell cycle. Okay, so this is one difference between the two. And in the meiosis 1, the end product, that is the final cell number. Is it audible? Uh, can anyone suggest, like, is it audible currently? Hello,
Yes,
is my
sir.
screen? Yeah. Okay. Thank you. Yeah. So the sister chromatids are separated. Uh, okay. So that was what was I was discussing, that in the meiosis two, the end product is the haploid cell, but in meiosis one, it is a diploid cell end product. Okay. And meiosis two resembles the mitosis, but the sister chromatids actually after the end product are different. Okay. You can here you get four haploid cells after the meiosis two. So this is the prophase stage of the meiosis one. Okay, the meiosis one prophase stage are further divided into the five stages, and actually the prophase stage is the important stage in the cell cycle of the meiosis, in which there are at the different stages um, different important things occur, which helps in the genetic information exchange. So there are five different stages: leptotin, zygotin, pectitin, diplotin, and dikinesis. So in the leptotin, generally the chromatin gets condensed, because in the synthesis stage earlier, when the DNA was replicated, the chromosomes are generally in the loosed form. Okay, it is in the decondensed form. So during the prophase leptotin stage, that is the initial stage of the prophase, the chromatin gets condensed, and the nuclear membrane start to get disappeared. Okay, it start to degrade at this stage slightly. Then in the zygotin stage, the homologous chromosomes gets nearby to each other. Okay, the two non-sister chromatids, that is, which are homologous to each other, that is, the genetic information almost are similar between each other, occurs in the bivalent form, and it forms a bivalent, that is, the two homologous chromosomes pair with each other, and that is called bivalent. Okay, and the synaptonomal complex formation starts to occur. So synaptonomal complex is a complex by which the two homologous chromosomes pair with each other. Okay, so that is called synaptonomal complex, and there are various proteins involved in between the synaptonomal complex formation that we'll be discussing further. Okay, and after the zygote stage, here you can see the nuclear membrane is starting to disappear in the zygote more compared to lactote. Okay, and in the pectine stage, the synaptonomal complex again start to is formed between the two bivalents, two homologous chromosomes. And here you can see the exchange of genetic information. So in the pectine stage, the crossing over occurs and the genetic information are exchanged between the two homologous chromosomes. Okay, so that crossing over occurs in the pectine stage, and in the diplotin stage, the crossing over has been done. Okay, and synaptonomal complex start to disappear. This synaptonomal complex between the two homologous chromosomes disappear between them, and the crossing over has also occurred, but the two homologous chromosomes remains attached to each other by the chiasma. So chiasma is the place where the two homologous chromosomes genetic information has been exchanged between the two homologous chromosomes. So the chromosome remains attached by the chiasma, and in the dikinesis stage, the attachment gets disrupted from there as well, and the two homologous chromosomes separate through each other, and the nuclear membrane completely dissociates. So the disappearance of nuclear membrane is important because then only the spindle fibers can attach to the centromere of the chromosome. Okay, so in the dikinesis stage, the nuclear membrane completely disappears, and the two homologous chromosomes are separated through each other by the from the chiasm. So these are the five important stages in the prophase, and you need to remember this stage. Okay, each and every stage. So as I discussed that the synaptonomal complex is important. Okay, so synaptonomal complex is formed between the two homologous chromosomes, and it is important to form the chiasma. If there is a synaptonomal complex is not strong enough, then the genetic information exchange between them, it will lead to disruption in this crossing over. Okay, during the crossing over stage, it will lead to its disruption, and it will not be proper. So synaptonomal complex is important in that regard, and to bind the two homologous chromosome between them. So the during the how the synaptonomal complex is formed, here it is shown. So there are three key elements. One is central element, another is transverse and lateral element. So these are the chromatin loops. Okay, these are the chromatin loops, and here you can see the lateral elements, which is made up of HOP1 and RAD1 protein. Okay, so these lateral elements are made up of HOP1 and RAD1 protein, and in the middle, you can see the proteins are ECM11 and GMC2. So these are the proteins which anchors the two homologous chromosome by the anchors the central element by the with the lateral elements. Okay, this protein, and the zip one acts as an anchor between the central and the lateral element. So that is a transverse element. Okay, and this central element binds the lateral elements by with the two chromatin, and that is how the two homologous chromosomes are paired with each other intactly. Okay, and during the dissolution of the synaptonemal complex, the these protein elements are disrupted. The linkage between these protein elements are disrupted, and there you can get the two homologous chromosomes get separated if this linkage is disrupted. Okay, so this is the proteins which are involved in the synaptonemal complex formation. Here we are talking about Saccharomyces cerevisiae, okay, which is a budding yeast. So these are the proteins which are involved in that organism. So here you can see the detailed stage wise of the meiosis, prophase one of the meiosis, stage wise the synaptonemal complex formation and the disassembly. So in the lactotin stage, as we discussed, that the two homologous chromosomes come closer to each other. In the zygote stage, the synaptonomal complex assembly start occurring, At the peg-chitin stage, the complete synaptonemal complex is formed between the two homologous chromosomes, and the genetic information are exchanged between the two homologous chromosomes by crossing over. In the diplotin stage and in the dikinesis, the synaptonemal complex gets disassembled, and these two sister chromosomes, two homologous chromosomes, are separated by each other. Okay, so this is the here we are discussing the dynamics of the synaptonemal complex formation. So peg-chitin stage is a stage where the complete synaptonemal complex is there, and in the diplotin stage, the synaptonemal complex get disassembled. So why it is important for the synaptonemal complex to form properly, okay, and at the correct time, it should get disassembled as well. If there is any problem in this in any of these stages, then that it will be leading to the non-disjunction of the chromosome. If the synaptonemal complex is not strong enough to separate the two homologous chromosome, or if it is not if it does not get disassembled, then there will be a genetic error, okay, genetic abnormalities in the individual because of the improper chromosome segregation. And we know that if the chromosomes are not segregated properly between each cells, then there are various genetic diseases which occur, and some of them we'll be discussing further. Okay, but the dynamics of the synaptonemal complex formation and disassembly is important for the proper segregation of the chromosome. Here you can see different types of the non-disjunction formation. So in the normal meiosis 1 stage, in the normal stage, the two homologous chromosomes are separated by each other in the meiosis 1. Okay, but when it is a non-disjunction, when we term that is a true non-disjunction, over there the two homologous chromosomes, the synaptonemal complex between the two homologous chromosomes does not get disassembled in the diplotin stage, and due to the non-disassembly of the synaptonemal complex, there is a non-disjunction, and the homologous chromosomes get transferred to the single cell, and in another cell there will be no chromosomes present. Okay, so in one cell there will be a 4n number of chromosome. Okay, and another cell there will be no chromosome present. So this is a process of true non-disjunction. In the achiasmatic non-disjunction as well, the one sister chromatids of the one arm and another sister chromatids get separated in a single chromosomes, but before crossing over does not occur in this stage. Okay, so that is called achiasmatic non-disjunction, because the chiasma formation, which is a which provides the genetic variations, that doesn't occur in the achiasmatic non-disjunction, and before the chiasma formation and the synaptonemal complex formation, the separation occurs. Okay, so synaptonemal complex formation and chiasma formation go hand in hand. Okay, so if both of the stages does not occur and then directly the chromosomes get separated, then there will be also be a problem. Okay, the genetic information will not be shared between the two sister chromosomes, two homologous chromosomes. So that is called achiasmat non-disjunction. That is, without formation of chiasma, the separation occurs of the homologous chromosome. In the premature separation of chromatids, as you can see in this example, uh, the one homologous chromosomes as well as the one sister chromatids of another homologous chromosomes get transferred into a single cell, and one single sister chromatids get separated into another cell. Okay, so in one cell there will be 2n plus 1 phenotype, and another cell there will be 2n minus 1 phenotype. So 2n means two sister chromatids, and minus 1 means one sister chromatids is missing. 2n plus 1 means one sister chromatids is extra in that cell. So that is the phenotype when we talk about, okay, regarding the chromosomes. And we already know the genetic effect of this a non-disjunction. There are various phenotypes and the disease currently we observe. Some of them are most common are the Down syndrome, in which there is non-disjunction. Okay, Turner syndrome, as well as the Klinefelter syndrome, in which there is one extra sex chromosomes, chromatids. Okay, one extra sex chromatids are present. So this all are due to the non-disjunction in the sister chromosome during the separation or during the meiosis. So this we talked about the meiosis 1, and in meiosis 2 as well. Here you can see the normal separation is between the two sister chromatids of a single chromosome. But in non-disjunction, generally you get one single chromosome separated into one cell and another cell. There is no chromosome present. Okay, so that is occurs in the meiosis 2 stages. So these are the complete non-disjunction formation in the meiosis 1 and 2. Is it audible, clear to everyone till this stage? Are you clear with the meiosis stages? So here, this is a summarized version of the meiosis. Okay, meiosis stage 1 and 2. And here you can see meiosis. There is the main purpose of meiosis is the genetic variation which occurs during the chiasma formation and maintenance of ploidy. Okay, so maintenance of ploidy means the each and every cell should have the haploid number of chromosomes. If there is a diploid number of chromosome after the meiosis 2, then during the fertilization, when you get a haploid chromosome, then you will get one extra chromatid. Okay, and that will lead to disease condition. Okay, just a second. Yeah, sorry for the disturbance. Is it audible to everyone? Can anyone confirm? Yeah. So yeah, thank you. So in the meiosis stage, as we discussed, that genetic variation is there. The main purpose of meiosis is to have the genetic variation and to maintain the ploidy level in the cell. Okay, so that each and every cell contains a haploid number of chromosome, and not the diploid. And if it is abnormalities is there, then separation during the chromosome, then it will be a problem. So this is the stage G1 as G2, and in meiosis 1, here you can see prophase 1, metaphase 1, anaphase, where the homologous chromosome segregation occurs, telophase and cytokinesis. And prophase 1 is again separated into leptotin, zygotin, pectidin. In the pectidin stage, here you can see synaptonemal complex crossing maturation and crossing over occurs. In the diplotin stage, the synaptonemal complex get disassembled, and then there is a in the dikinesis. Generally, the separation occurs between the homologous chromosome. In the meiosis 2, again the same stages are there. The difference in the anaphase 1 and the anaphase 2 is in the anaphase 1, the homologous chromosomes get separated, and the final form cell after the meiosis 1 is the diploid cell. But during the anaphase 2, the sister chromatids is segregated, and the final after the meiosis 2, the final product, the final phenotype of the cell is haploid. Okay, so this is the thing. And now we'll be talking about the homologous recombination. So homologous recombination is an important stage during the cell cycle, during the recombination process, as we discussed the formation of chiasma and the genetic information occurring between the two homologous chromosomes. So during that phase, by the process of homologous recombination, only the genetic information forms between the two homologous chromosomes. Okay. So homologous recombination generally generally occurs in the cells during the two processes. First one is during the meiosis stage only. During the meiosis stage, where the preferred template is the non-sister chromatid, homologous non-sister chromatid. Okay, and during the DNA repair as well. In the mitosis, we studied DNA repair checkpoints, and if there is a double-stranded break in the DNA, various proteins were getting activated. Some proteins, as you can recall, are ATM and ATR protein, okay, which activates the P53, and that P53 act as a repressor of the S-phase cyclins gene. So at that stage, if the DNA is damaged, the DNA during the DNA repair process, the homologous recombination occurs, and the sister chromatids are by the template is the sister chromatid. Okay, so these are the two ways why homologous recombination. Then in the homologous chromosome recombination, generally at the initiation stage, the formation of double strand breaks occur first. Okay, and then uh, first the formation of the double strand break occurs, then the recombinogenic single-stranded DNA tails is formed. So single-stranded DNA is formed after the double-strand break. The single-strand DNA tail is formed. Okay, that is called double-strand break processing. And then that single-stranded DNA uh, finds with the homologous chromosome, and there then the DNA repair occurs during the by the using the homologous searching. Okay, that is called homolog searching by the different the other other homologous chromosomes which are present in its vicinity. So that single-stranded DNA search for this homolog, and then the homologous recombination resolution occurs. Okay, so that is why. So in this example here, you can see let's discuss by through this diagram, so it will be more clear to you. So in the leptotin stage generally, so we will talk stage wise as well. In the leptotin stage, the homologous chromosomes are the DNA double-stranded break. Here you can see the DNA double-stranded break occurs, and the end resection occurs. End resections are the formation of single-stranded DNA tail. Okay, so this single-stranded portion of this DNA then translocate to the other homologous chromosome, the other pair of the homologous chromosome. Okay, so that is why the single strand formation of the DNA is important. Otherwise it will not be able to form the homologs. So these are mediated through different proteins. SPO1 is one of the topoisomerase. It helps in unwinding of the DNA and breakage of this double-stranded break, do the double-stranded break. Another protein is RAD50, which helps in the formation of the single-stranded DNA molecule, and some other proteins such as RAD51, SAE3, HOP2, MND1, okay, which helps in the during the invasion of the single-stranded DNA into the other homologous chromosome, and then the homologous searching occurs. Okay, so it interacts with this homologous chromatid in the leptotin stage, and after that, after the homologous searching, here you get two products. One is like crossover pathway, and another is non-crossover pathway. So this homologous chromosome go through both this pathway. Okay, one of these two pathway basically crossover as well as non-crossover. So in the crossover pathway, let's discuss first about the non-crossover pathway. Okay, the idea by the non-crossover pathway is that in the non-crossover pathway, the DNA which was break and it was searching for the homologs. It does after the homologs have been formed. Okay, after the DNA has been synthesized of this breakage, after the breakage, the resolution occurs, and the homologous chromosome does not become part. Okay, the other pair of the homologous chromosome does not become part of the chromosome or this chromosome. Okay, so the but in the crossover pathway here you can see the other pair of the chromosome also become part of the sister chromosome. So that you can see the genetic information occurring. So over here you can easily guess that if the crossover pathway is generally preferred in the meiosis. Okay, but the non-crossover pathways are generally preferred during the mitosis, during the DNA repair process, because in the DNA repair generally if there is a double-stranded break, then we only want that part of the DNA it searches for the homologs, okay, and it synthesizes the DNA, but the genetic information doesn't occur. So in non-crossover pathway, genetic information exchange of genetic information doesn't occur, and that's what actually we want. We don't want genetic information should be exchange in the during the DNA repair process, okay, apart from meiosis. But in the meiosis stage, we need the exchange of the genetic information. So there you need the crossover pathway to form. Okay. So now in the non-crossover pathway, the DNA is synthesized, and here you can see after the DNA is synthesized, the complete DNA is synthesized and ligation occurs in the non-crossover pathway. But the both the chromosomes, both the homologous chromosomes have the individual genetic information. The genetic information has not been exchanged between them. Okay. But in the crossover pathway, here you can see there is a formation of this crossover. Okay, by certain proteins. Here you can see zip1, zip2, zip3, mol3, and MSH5. And the crossing over occurs. Here you can see the crossing over product. In the zygoting stage, generally the crossing over occurs. Okay. In the pectitin stage, the crossing over DNA synthesis has done, and the DNA has been ligated by another crossover product. So here you can see two crossovers, one at this stage, and another is at the this stage of the chromosome. So two crossovers is there, and then there is a resolution, and this is actually called the holiday junction. Okay, the crossover you can see two crossover here you can see of the one pair of chromosome is called a holiday junction, and after that the resol during after the resolution here you can see the exchange of genetic information between the chromosomes in the diplotin stage of the cell cycle. Okay, so this specifically crossover pathway specifically occurs in the meiosis stage because we need exchange of genetic information which occurs at the point of chiasma formation. The chiasma generally this crossover occurs and we get exchange of the genetic information. But during the DNA repair pathway in the normal cells, the non-crossover pathway are generally formed. Okay. So these are the information by the crossover and non-crossover regarding the crossover and non-crossover. And in the meiotic prophase stage, generally the homologous recombination occurs and it leads to the genetic variation and survival. Okay, uh, of the individual in the individual. So genetic variation, as we already discussed that about the genetic variation. Okay, but the in the better survival, how this homologous recombination in the meiosis helps in the better survival, we'll be discussing it further. There are various regulations happening, okay, and that leads to the better survival of the homologous in the during the meiosis by the meiosis, okay, process. So as I discussed that proper segregation of chromosome are important, and if it does not lead to proper segregation, then here you can find various conditions such as aneuploidy. Aneuploidy is the means the number of chromosomes which should be present in the cell are less. Okay, uh, uh, the ploidy is different in that cell, so that is called aneuploidy. The chromosomes is not present, and here you can see different diseases: Turner syndrome, Down syndrome, Klinefelter syndrome. So in Turner syndrome, generally the instead of the two X chromosome, there is only one X chromosome present. Okay, two X chromosome in case of female and X and Y chromosome in case of male. So instead of two sex chromosomes, only one sex chromosome is present, and that leads to the Turner syndrome. So the ploidy level is XO, okay, n minus one XO chromosome is present. Only one X chromosome is present. In Down syndrome, generally one chromatids are more in the 21st number of chromosome. Okay, so the ploidy level is 2n plus one in the Down syndrome. So there is one extra chromatid which is present, and in the uh, Klinefelter syndrome as well, there is one extra sex chromosome which is present. Okay, there are two X and one Y chromosome. So due to that, there is abnormalities in the individual because of the extra X chromosome present. And during the homologous pairing, generally, there is this in the Saccharomyces pombe and fungi and all those things. There are different pairing occurs. Okay, in the female Drosophila and C. elegans, here you can see pairing is independent of synapses. So pairing occurs, but the synaptonomal complex does not form. In the Fijian yeast, that is Saccharomyces pombe and Aspergillus as well, recombination mediated pairing occurs, but the synaptonomal complex is absent. So in certain mammals, in the certain organisms, without the formation of synaptonomal complex, you can see observe the pairing. Okay, pairing occurring between the two sister chromosomes, between the two homologous chromosomes. That pairing actually occurs by the centromere. Okay, between the centromere, the two homologous chromosomes are linked to each other, and then the pairing occurs. In the mammals, generally, in the Saccharomyces cerevisiae, as we discussed, there are the synaptonomal complex are formed throughout the chromosome, but in certain individuals or certain organisms, the pairing is synaptonomal complex is formed at the centromeric region only. Okay, not throughout the whole chromosome. So that is the reason. That is the variation which occurs. So this is the summary version, like for regarding the homologous recombination. That is double-stranded break if it is formed, then a single-stranded tail is first generated. Then the strain gets invaded. Okay, single strain invasion occurs, single-stranded tail. Then there is a resolution, that is crossovers and non-crossovers. And no crossovers are generally preferred in the meiosis stage. Non-crossover occurs during the DNA repair stage, and it leads to the homologous pairing, segregation, and variation. And homologous pairing, double strand break independent mechanism, there can occur by the centromere mediated. That is the between the centromere, the synaptonomal complex are formed, and then the homologous pairing occurs between the chromosome. It can be telomere mediated. That is at the end of the chromosome. That is at the telomere stage, the synaptonomal complex is formed, not throughout the chromosome. Only at the telomere stage the chromosomes synaptomal complex is formed, and then there is a exchange of information in between. Okay, chiasma formation is there. In the RNA mediated, there are some RNA, okay, which helps in the synaptomal complex formation, okay, and that helps in the chiasma formation and exchange of genetic information. So this is regarding the homologous recombination process. Is it clear to everyone? Is it clear to everyone up till here? Is there any doubt? Thank you. Yeah, so let's discuss some questions uh, regarding the assignments. Okay, so uh, first question is the differential localization of RAN GTP and RAN GDP in the cell compartment is important because it helps in the formation of nuclear pore complex. It provides directionality to the nuclear input, helps in recognition of the nuclear localization signal, and none of the above. So RAN GTP and RAN GTP are helpful in the uh, transform localization of protein from the cytoplasm to the nucleus, okay, and vice versa. So there is a GTP and GTP. RAN protein is a nuclear receptor, okay, nuclear protein, which is helpful in that regard. So the correct option is it provides directionality to the nuclear input, okay. So RAN GTP, here you can see that in protein alpha and beta are bounded to this protein TRPS1, which is a nuclear protein, and after binding to it, it translocated to the nucleus, and over then the RAN GTP is converted. Okay, RAN GTP by the help of the RAN GTP, the protein is translocated inside the nucleus, and then the RAN GTP complex is again transferred into the cytoplasm, and where it is GTP is converted into again into GDP by the GTPase activating protein. Okay, gap protein, and the RAN GTP is regulated by the conine exchange factor. That is how it helps in the nuclear import and export process. The second question is which of the following option correctly describe an event occurring in the prophase 1? Okay. So during the prophase 1, which of the following options correctly describe the prophase 1 of meiosis? So pairing of homologous chromosome and crossing over, alignment of homologous chromosome, duplication of DNA, and segregation of homologous chromosome. So during the prophase 1, generally the pairing of homologous chromosome and crossing over occurs. So this is regarding the prophase 1. Okay. And here you can see the homologous chromosomes are paired with each other during the prophase 1. Third question is pick the correct order of the different stages in the meiotic prophase. So which is the correct option? Correct option is option A, leptotin, zygote, pectin, diplotin, and dikinesis. This is the correct option. Okay, these are the stages of the prophase. So here you can see leptotin, zygotin, pectin, diplotin, and dikinesis stages are there. Fourth is synaptonemal complex disassembly takes place in which stage? So synaptonemal complex disassembly, as we discussed, it occurs in the diplotin stage. Okay, so option B is correct. During the diplotin stage, the synaptonemal complex gets disassembled. Fifth is non-disjunction due to improper segregation may lead to aneuploidy, Down syndrome, progeria, and none of the above. So the correct option is there are two correct options: aneuploidy and Down syndrome. So these are the two correct options which occurs due to improper segregation of chromosome. Progeria generally occurs. Progeria generally occurs due to lamin protein. This mutation in the lamin protein, progeria occurs. Okay, so these are meiosis, non-disjunction as we discussed. Uh, During homologous recombination, the point at which the chromatid from a pair of homologous chromosomes are physically attached to each other is called it is called chiasma. Okay, the chiasma is a point at which the pair of homologous chromosomes are physically attached to each other. In budding yeast, the meiotic commitment initiator IME1 act as a the correct option is transcription factor. Okay, so in the budding is generally in the regulation of meiotic G1S phase. In the budding is when the nutrient is depleted. Okay, there is when there is a nutrient deprivation, there is an expression of regulators. So during the nutrient deprivation stage, the cells undergo the meiosis stage. Okay, cells undergo the meiosis, and at that stage, generally the IME1 protein is expressed. It targets the IME2. Okay, and helps in the transcription of IME2. So IME2 is a transcription factor which helps the encodes the meiosis specific kinase and helps in the activation of specific CDK and cycling, specific cycling which help in the uh, meiosis process. Okay, so correct option is this transcription factor. The proteins that are targeted to nucleus have a signal sequence that are reaching positively charged amino acids. Okay, option A. So the nuclear localization signal generally has a positively charged amino acids, that is lysine and arginine. Okay, to which the nuclear receptor binds, and then the proteins are translocated into the nucleus. So pick the correct statement regarding the nuclear organization. Heterochromatin region is darkly stained and transcriptionally active. Euchromatin is lightly stained and transcriptionally active. Histone deacetylation by SIR2 causes euchromatin formation. So the correct option is option B. Euchromatin is lightly stained and transcriptionally active. So heterochromatin are darkly stained generally, and it is transcriptionally repressed region. Okay, whereas euchromatin is transcriptionally active region, and it is lightly stained chromatin. And histone deacetylation by SIR2 and HTAC generally promotes the heterochromatin formation, okay, not euchromatin. So this is the case. The cytoskeletal supporting the nuclear membrane is made up of which protein? So it is made up of lamin protein. Okay, so lamin dimers are there, lamin polymer. So it ties the nuclear membrane, and that helps in the formation of the cytoskeleton, the nuclear membrane. Okay, so that's it from this lecture, and we can discuss further in the next lecture. Okay, thank you everyone.